All right. Good afternoon. My name is Cornelia Polyak, and today is a bittersweet announcement, as you've seen on the electronic announcement. Also, today is the last Connect Science seminar. So I wanted to join to thank everyone for running this seminar series. So all of us who have been participating are here, except David Weinstock. So just a quick reminder, we started this seminar series in March 2020 when we had the shutdown due to the pandemic. And initially we were running it with, David Weinstock, and then Richard Oakley, who's on here, and Rosemary Nabukera, who is also here on the screen. And the four of us were pretty much organizing, and Richard and Rosemary took care of all the logistics, and David and I were doing the hosting. Then as life was becoming more normal, we switched to twice a week and then once a week. And then Claudia Steele, who's right next to me, she took over the logistics of inviting speakers and handling the setup. And it's been really successful. Many of you and many of the attendees have sent me emails how much they enjoyed the seminar series, and it's been very well attended. So we're very happy that we had this contribution to the scientific community. And but right now many of our speakers prefer to be in person, and we prefer to be in-person seminars that gives more personal interaction. So we thought that after more than 3 years of running, it will be time to discontinue. All the recorded seminars still remain on the website, so you can go back and listen to the speakers. And as you know, we had really, really great speakers around the world, including today's speaker as well. And many seminars have been extremely well attended. So thank you again. Thank you for Richard and Rosemary and Claudia for hosting these seminars and taking care of all the logistics. And thank you everyone for participating and attending and showing up to these seminars. So I just want to say bye. And now I hand it over to Sandy McAllister.
Thank you. I also want to add a special thank you to Nellie. This has been a phenomenal seminar series, not only to help get us through the pandemic and still feel connected at a time we really needed it, but ever since then, over the past 3 years, this seminar series has had a real impact on the world and enabled people to attend and engage in scientific discussion that might not otherwise be able to hop on a plane. So thank you, Nellie, for all your efforts and keeping us going over the past few years. Thank you. A few housekeeping notes before we begin. Please remember to post your questions to the Q&A and not the chat. That way I can read them at the end, and any questions that we're unable to get to, we will they're often sent to the speakers for further engagement offline. So with that, it is my truly distinct honor and pleasure to introduce Dr. Judy Varner today. She's not only a pioneer in our field of tumor microenvironment and tumor immunology, but also a dear friend and colleague with whom I have the pleasure of working. Dr. Varner is a professor at UCSD in the departments of pathology and medicine. And she's also the director of the Moores Cancer Center Solid Tumor Therapeutics Program and recent director of the faculty mentoring program in the Department of Pathology. She also is a co-founder of another phenomenal seminar series, the Myeloid Network, which is an international network of researchers studying and targeting macrophages, granulocytes, and dendritic cells in health and disease. Dr. Varner's research is focused on targeting macrophages and the role of macrophages and other myeloid cells in cancer and chronic diseases. She's identified novel mechanisms by which macrophages promote tumor growth, and she's really developed amazing new approaches to treat cancer patients by targeting macrophages and signal transduction in myeloid and macrophage cells, including an investigational drug, a PI3K gamma inhibitor, eganelisib, which hopefully we'll hear more about today. Her work has appeared in all of the top journals: Cancer Cell, Nature Communications, Cancer Discovery. And among her list of impressive awards, I'll include that she is a recipient of a CureBound Foundation, Immunotherapy Foundation, a V Foundation, an AACR Landon Foundation, and Lustgarten Foundation awards for her research. So with that, I will turn it over to Dr. Varner. Thank you for joining us today on this special day.
Thank you, Sandy and Nellie, for this fantastic invitation. I'm honored to speak to you, and especially to be the last speaker in your series. Having started a seminar series during COVID that's online that reaches the international community, I can really appreciate the value of these series. So perhaps you can find a way to continue some form of an online seminar series in the future. So what I want to tell you about today is our research into figuring out how to alter cancer immune suppression, and to tell you about the basic research studies we've done that have led to successes in the clinic. So, of course, none of my work could be done without the valuable contributions of postdocs who are dear collaborators and colleagues now, including Michael Schmidt, Megan Kaneda, Paulina Pathria, Hide Takahashi, Angusen Gebremedin, and Alice Chen. So you'll hear about their work coming up. So these are my disclosures. So we know that tumor immune systems, our ecosystems, are really complex. There's a whole variety of cell types in a tumor besides tumor cells. We have T cells, which typically are immune suppressed, regulatory T cells, CD4 T cells that are not activating killer T cells, regulatory B cells, and other cells, but particularly most abundant are macrophages. If we had good anti-tumor responses, we would have killer T cells, NK cells, and antibody-producing cells that would be working effectively. But unfortunately, that's typically not the case. So the way we know that tumors are complex in terms of their immune systems are through work like this from Lisa Cousin's lab, where they've done multiplex immunohistochemistry through a very clever strategy of incubating tissues with sets of antibodies, removing the antibodies, and then staining with additional antibodies and co-registering all of these markers. So by doing this kind of approach, you can see that there are numerous cell types that can stain with different markers for T cells and myeloid cells. And single cell sequencing and other strategies have taught us that there are actually diverse types of cells and diverse states in the tumor. So how do we approach this? Well, first of all, we know that a key goal of removing tumors is to have activated CD8 positive T cells or NK cells that produce granzymes and perforins that can actually kill tumor cells. And there are some tumors that are full of CD8 positive T cells, and they're just not quite as active as they should be because they are exhausted. And so we have seen the development of checkpoint inhibitors that have been really effective in these particular tumors. Then there are tumors where there are many T cells, but they're all at the boundary of the tumor, excluded by the presence of macrophages and fibroblasts that keep them from penetrating. These are less responsive to checkpoint inhibitors. And then we have those immunologically ignorant tumors which have no apparent T cells present. So how do we deal with these two types of tumors that are non-responsive to T cell therapy? Well, immune therapy has been in existence for a long time. Since the very first smallpox vaccine was used and other vaccines were used in attempts to manage tumors, including the BCG vaccine that is in some ways still used today. And here's the timeline of the numerous attempts to develop immune therapy. And even the Nobel Prize was awarded for development of such therapeutics. But we still don't have the ability to control the immune suppressive tumor microenvironment in many patients. And so we need to rethink whether activating T cells is sufficient. And those of us who work on myeloid cells believe that these are the cells that must be targeted in order to fully activate the anti-tumor immune response. So myeloid cells are comprised of a whole variety of cells like mast cells, dendritic cells, macrophages, neutrophils, eosinophils, and basophils. You can find these cell types in every tumor in different proportions depending upon the tumor. And they are ordinarily in an immune response, in a natural immune response, able to interact with the adaptive immune system and to work in a coordinated fashion to eliminate infections and provide wound healing. As you can see here, this beautiful image of a macrophage attacking bacteria would be an ideal representation of how a macrophage works. Macrophages also play important roles in laying down networks of cells during embryonic development, including blood vessels, as seen here. And that is one of the problems with macrophages, that they normally do promote angiogenesis and cell migration as part of their wound healing response, which is exaggerated in tumors. So in tumors, we see an overabundance of macrophages stained here with anti-CD68 in this human pancreatic ductal adenocarcinoma tissue section, while a normal tissue has relatively few macrophages. So in fact, macrophages can make up the majority of the immune cells in a tissue, and they have been associated with poor survival. Here we have the association of CD68 positive staining with poor survival in patients with pancreatic cancer. So we must contend with macrophages and try to understand what they're doing. So macrophages can take on several different kinds of states, but we've all heard there are two basic states. There's the pro-inflammatory that produces factors such as nitric oxide and reactive oxygen species, tumor necrosis factor, and IL-1 beta, and they are clearly associated with pathogen clearance. They also produce factors such as complement and cytokines that can activate and recruit T cells. However, macrophages can also be wound healing in their state. They can produce cytokines that stimulate a Th2 helper T cell response and suppress immune cells. So they are designed to shut down an over exuberant immune response when pathogens have been cleared and to promote wound healing. Unfortunately, this is the type of macrophage that tends to be found in tumors, and there are a lot of reasons for that. Here is some examples of studies that we have done that can show you how significant the macrophage situation, the myeloid cell situation, is in a tumor. Here we have examined the presence of myeloid cells in normal tissues and tumor tissues by CyTOF, time of flight mass cytometry. And in red, you see all myeloid cells. So after, in a normal tissue, there are some resident myeloid cells, also some passing through blood vessels. Immediately after implanting tumor cells, we have a huge accumulation of myeloid cells, primarily bone marrow-derived monocytes and granulocytes. And then as time goes on, they just continue to accumulate until they comprise the majority of cells in a tumor. These cells are associated directly with the increase in blood vessels in a tumor in almost a linear fashion, as they produce vascular endothelial growth factor and other factors that promote angiogenesis and help a tumor to grow. So years ago, we asked whether we could target myeloid cell accumulation to suppress tumor growth. At that time, most people thought most myeloid cells were in fact bone marrow derived, And on recent days, we now realize some myeloid cells are resident within tissues. So we asked if we can target trafficking or now proliferation of resident cells, can we actually cause a better immune response that could then interact with CD8 positive T cells and eradicate tumors? So Michael Schmidt, a postdoc in the lab back in 2011, asked how do myeloid cells get into tumors? We had known through work in the immunity field that myeloid cells and other cells, such as T cells, circulate through the blood system with cell adhesion proteins that are kept in an inactive state. They're folded toward the membrane, and it requires a signal transduction cascade from a neighboring cytokine, chemokine, or other G protein coupled receptor ligand, such as thrombin, to cause integrins to unfold and interact with their ligand, which in this case would be the endothelial cell. So Michael first identified that the major cell adhesion protein that facilitated entry into tumors was the integrin alpha 4 beta 1. Other integrins did not appear to be regulating this process. And then he asked, wow, what were the signals that activated this integrin? And he found that numerous signals, cytokines, chemokines, that were known to be involved in tumors could activate the integrin. So he reasoned that there must be a common signaling cascade downstream of these that could activate the integrin and promote tumor inflammation. He screened a number of different inhibitors and found, interestingly, only a single pathway was regulating this process, and that was the PI3 kinase pathway. In particular, he devised an assay, for example, this one, where he allowed myeloid cells to attach to vascular endothelial growth factor, which is the protein on endothelial cells that mediates the trafficking of myeloid cells into tissues. And he found that inhibitors of PI3 kinase alpha, beta, or delta had no effect on this. But inhibitors of PI3 kinase gamma completely shut this process down. PI3 kinase gamma knockout myeloid cells also couldn't adhere. And inhibitors of PI3 kinase gamma were very effective at inhibiting all the adhesion induced by these stimuli. So he also showed them that these inhibitors could prevent the activation of AKT, which is what you would expect to be activated by PI3 kinase in cells. So he was able then to determine that PI3 kinase gamma was a major isoform in myeloid cells. So by doing this kind of immunostaining for different PI3 kinases, he found that macrophages and tumor-associated macrophages mainly express gamma, not other PI3 kinase isoforms, while B cells and T cells mainly express delta, which is regulating their trafficking, and tumor cells mainly express alpha and beta. So interestingly, PI3 kinases, alpha is expressed in myeloid cells, but to a much less extent than gamma. So these are two different classes of PI3 kinases. Gamma has a unique structure compared to alpha, beta, and delta because it lacks the ability to be activated by P85, which is a SH2 binding protein that's commonly activated by tyrosine kinase receptors and chemokine receptors. So, not chemokine receptors, but PI3 kinase gamma was well known to be activated by chemokine receptors. We showed that it had to be activated strictly by interacting with RAS, and numerous types of signals can activate RAS. And we showed that numerous signals can activate PI3 kinase gamma. So its structure has long been crystallized, and numerous inhibitors have been developed. And now there is one inhibitor that's well advanced in clinical trials for cancer that I'll tell you about. So we showed that PI3 kinase gamma is expressed in macrophages in different tissues. And so here we have a staining of actually COVID lungs that have been stain for p-kinase gamma and CD68. And these are serial sections, and we've been able to show through double staining that the same cells are staining for gamma and CD68. So it's a major marker of myeloid cells, and it plays a great functional role in inflammation. We've done a number of studies in inflammation models to show that p-kinase gamma is essential for trafficking toward inflammatory stimuli. So this is the work of Angus Gebremedin, a postdoc in the lab, and Ryan Shepard, former technician who's now a graduate student at UCSD. So what we did was we asked whether common inflammatory stimuli like thioglycolate injected into the peritoneum could elicit macrophage accumulation. And here you can see that it is a very potent elicitor of macrophage inflammation compared to normal peritoneum. And you can see that very much reduced accumulation of such cells in p-kinase gamma knockout animals. We also found that this IPI 549, again, elicit, was able to similarly reduce the trafficking of myeloid cells into the peritoneum induced by thioglycolate. We then have gone on to show that inflammatory stimuli such as lipopolysaccharide or MRSA, E. coli, and even COVID could induce inflammation in wild-type animals, but not in p-kinase gamma knockout animals or drug-treated animals. So there's a substantial inhibition of recruitment of myeloid cells and macrophages and neutrophils into these tissues. And in fact, there's a improved survival when animals have been treated with a lethal dose of lipopolysaccharide. Interestingly, there's a better survival for males than females, which have a much more robust anti-inflammatory response than males. We also found an improved survival if we inoculated mice with lethal doses of methicillin-resistant Staphylococcus aureus, a really large clinical problem, or drug-resistant E. coli. So essentially, in a number of papers that we published over the years through the work of Michael Schmidt and Megan Kaneda, we've been able to establish this mechanism. A variety of different receptors can activate PI3 kinase gamma by activating RAS. This draws PI3 kinase gamma into the membrane, and then it can activate this cascade of signaling processes that lead to the accumulation of RAP1 associated with TLN and paxillin on the integrin cytoplasmic tail. This recruits a high molecular weight form of myosin light chain kinase called MLCK210, which can then activate myosin and allow the integrin to attach to the actin cytoskeleton. We think this allows the integrin to unfold, and this happens within seconds or less than a second. In fact, this leads to tumor inflammation, and we've been able to show that inhibiting any of these molecules can suppress this active form of inflammation. But our research, so we have seen in animal models of cancer, such as this polyoma middle T mammary gland tumor model, in which animals develop hyperplasia, then invasive carcinoma over a period of up to 15 weeks. And we have seen that we observe inflammation, stained here with anti-CD11B, in the tumors of animals here with 9 weeks of tumor growth. At age 9 weeks, there's substantial hyperplasia and the beginnings of invasive carcinoma. But in the knockout animals or in drug-treated animals, there's very little inflammation. And in fact, what we see is a change in the nature of the tissue. The tumors are limited to hyperplasia. And even more surprising, we saw a strong survival benefit as although in our animal program, we were not allowed to do survival studies on these animals because they have such a large tumor burden. We were able to evaluate total tumor burden and observe that there's a significant reduction in total tumor burden in these animals, which develop tumor in all of their mammary glands. We did observe that there is a almost complete inhibition of metastasis when you inhibit PI3 kinase gamma in this animal model of breast cancer. We then also undertook with Megan Kaneda, the next postdoc in the lab, studies to determine whether we could inhibit pancreatic cancer development. We collaborated with our colleagues, Emilio Hirsch and Franco Novelli at the University of Torino in Italy, and who had crossed this KPC model with PI3 kinase gamma in knockout animals. And they were able to observe that there was a strong survival benefit if you deleted PI3 kinase gamma during KPC growth. You can see that there's a strong change also in the phenotype of the pancreas. While there's still hyperplasia in the PI3 kinase gamma knockout pancreas, there's a strong inhibition of invasive carcinoma, and there's a restoration of normal pancreas tissue. So we were able then to show that the incidence of invasive carcinoma was quantitatively inhibited. The normal acinar tissue was quantitatively retained, and the incidence of metastasis was also strongly suppressed. Most interestingly, we were able to strongly inhibit fibrosis in these models, both in the KRAS alone model and the KRAS P53 models. And that was also observed when we used an early PI3 kinase gamma inhibitor, TG100-115. This was also associated with an increase in the accumulation of CD4 positive and CD8 positive T cells within the tumors. So we went on to look at a variety of other implanted tumor models, including an HPV positive head and neck carcinoma, which shows remarkable effects. This model is immunogenic, and there are some spontaneous regressions, even in wild-type animals. But in PI3 kinase gamma knockout animals, we saw substantial inhibition of tumor growth. In a much more immune-suppressed tumor, a lung carcinoma tumor, we see at least a 50% inhibition of growth. And in orthotopic breast carcinoma models, we also see about a 50% inhibition of growth. We see similar effects when we use IPI-549, the clinical inhibitor, and we've seen this to be about the case in all of these different tumor models, suggesting that there is, potential wide-ranging benefit to inhibiting PI3 kinase gamma in patients. We've seen synergy with chemotherapy, but in particular, we've been asking, does PI3 kinase gamma only control myeloid cell trafficking? Because, somewhat to our surprise, when we look at some tumors through flow cytometry analysis or even single cell sequencing, we still see a substantial number of macrophages present in tissues. And we've begun looking at these to determine that these are actually resident macrophages that are remaining in the tumor. So why does PI3 kinase gamma inhibition actually work to inhibit tumor growth? So we asked whether PI3 kinase gamma could regulate macrophage states or phenotypes. And there's experimental systems where we can create bone marrow-derived macrophages by culturing monocytes from bone marrow in colony stimulating factor 1, which is a myeloid cell specific growth factor. And we can incubate them then in interferon gamma plus LPS to induce a pro-inflammatory macrophage, or in IL-4 to induce an immunosuppressive macrophage. And then we did RT-PCR for a variety of factors that are associated with each state. So interestingly, if we stimulate with IL-4 to make an M2 macrophage, we find that if we inhibit PI3 kinase gamma, we actually get strong stimulation of IL-12, nitric oxide, and IL-6, which are M1 or pro-inflammatory factors. If we stimulate with interferon gamma plus LPS, we also see a regulation of all of these as well as IL-1 and TNF alpha. That suggested to us we were repolarizing macrophages or preventing an immune suppressive macrophage state from occurring. We found also a strong stimulation of IL-12 beta secretion and a strong reduction of IL-10 protein secretion. This suggests that we have flipped the states of the macrophages to make them more pro-inflammatory. And then we did sequencing of the macrophages, and we determined, here I'm showing you the ratio of PI3 kinase gamma knockout macrophages to wild-type macrophages, and what you can see is there's a strong upregulation across the board of MHC molecules particularly, and as well as some other molecules, the fibronectin, which is associated with an immune response. Interferon gamma LPS macrophages particularly upregulate a variety of factors associated with a strong immune response. And then we see a strong downregulation of a variety of immunosuppressive factors as well as chemo attraction molecules that are associated with attracting myeloid cells into tissues. So there's a big changes that occur, and these tended to fall into these stimulation of immune response pathways, including T cell activation, adaptive immune responses, and so on. So Megan asked, how does this occur? And so she looked at the effect of PI3 kinase gamma inhibition on NF-kappa B. NF-kappa B is a molecule that's known to regulate the pro-inflammatory cytokine expression, particularly TNF alpha and others. And so she found, interestingly, that inhibiting PI3 kinase gamma not only stimulated the expression of the activation of NF-kappa B, but also extended its activation over up to 8 hours after stimulation. And she also found the same to be the case with the drug IPI-549. She then went on to show that there was an increase of NF-kappa B rel A binding to DNA, its target in DNA, and that indicates that the molecule was actually being activated and inducing gene expression. So through a series of studies, we realized that this was the other pathway that PI3 kinase gamma regulates. So ordinarily, it's regulating an immune suppressive response. So by activating AKT, mTOR, and S6 kinase, it then is able to activate CEPP beta in the absence of NF-kappa B, which promotes the expression of immune suppressive factors, such as arginase and other wound healing factors. Now, this occurs because it directly inhibits P65, NF-kappa B, and TBK1. But when we inhibit PI3 kinase gamma, the reverse occurs. We have the activation of TBK1 and NF-kappa B and a strong stimulation of gene expression. And we've been able to go on to show this occurs through epigenetic modeling of the chromatin structure, and we're currently trying to identify how that occurs. So Megan went on to show that this occurs in vivo. So by isolating either total tumor or macrophages from PI3 kinase gamma knockout in drug-treated animals, she was able to show that there's a downregulation of immune suppressive factors like arginase, IL-10, and TGF-beta, and an upregulation of nitric oxide synthase, IL-12, and interferon gamma within tumors. By doing RNA sequencing of whole tumors, we found that there was a strong signature of T cell activation and innate immunity. There's always an increase in CXCL9 and 10, which are known T cell chemoattractants. And these interferon gamma stimulated proteins, the guanylate binding proteins, which play roles in innate immunity. And then there's always an activation of both MHC class 1 and class 2 expression and a downregulation of immune suppressive factors. In particular, we observed a protein expression of MHC class 2 on tumor-associated macrophages in a variety of tumor types, which suggests that perhaps the major way this is working is by interacting with T cells to activate T cells. So we have recently been able to look at HPV positive tumors, and we found by protein expression, there's within PI3 kinase gamma knockout tumors, there's a strong upregulation of such these pro-inflammatory chemokines like TNF, IL-12, interferon gamma, and even IL-2. All of this suggests that PI3 kinase gamma inhibition in myeloid cells activates a strong T cell response. So we asked then the direct question, does it promote T cell activation? So Hideyuki Takahashi did some analysis of the quantification of T cells in tumors using this HPV positive head and neck tumor model. He found that there's a doubling of the percentage of CD8 positive T cells within tumors, and that these were largely activated effector cells by staining with markers for T cell memory and activated T cell phenotypes, and that they expressed an increased amount of granzyme B. So Megan then asked, does this synergize with checkpoint inhibitors? And would this then be a good therapeutic approach for patients? So we found that if we only treated with anti-PD-1 in this HPV positive models, we had limited effect. PI3 kinase gamma inhibition had a greater effect, but when we put the two together, they almost completely eradicated tumors and caused extended survival of animals. So Megan looked at what was going on within the tumors and found indeed there's an increase in granzyme B when you have this combination therapy, interferon gamma and IL-12. And there was a reduction in CD4 positive T cells, an increase in CD8 positive T cells. Interestingly, we found that anti-PD-1 had a stronger effect on MHC class 2, perhaps because of the upregulation of interferon gamma within these tumors. But clearly there's a strong immune response that's characterizing macrophages and T cells in this combinatorial treatment. So interestingly, we had many mice that survived for over 2 months. So we re-challenged them with tumor cells and found that whether they were PI3 kinase gamma knockout mice that had previously been treated with anti-PD-1 or inhibitor-treated mice, we found that there was immediate suppression of tumor regrowth. So Hideyuki Takahashi asked what was going on, and he looked within the spleens of animals that completely rejected tumors, and he found that there was a very strong presence of T effector memory cells in the knockout animals that was not present in the wild type animals. And that was also the case for CD8 and CD4 positive T cells. So there's a persistent immunological memory that's residing in the spleen of these animals. It's not present in the lymph nodes. And so it gave us potential to ask a few questions. So Hide did single cell sequencing of tumors at different time points to ask whether these effector memory cells were actually showing up within the tumors. And he found that if you looked at day 13 wild type versus day 20 wild type tumors, they have pretty much a similar appearance. But if you looked at PI3 kinase gamma knockout tumors at day 13 and day 20, he noticed something different. At day 13, there were a few more, there were some differences with wild type tumors, but at day 20, the differences were dramatic. there was a strong population of effector memory cells and effector T cells that was not present here within the wild type tumors. There were not particularly more exhausted T cells than in the wild type tumors. And there was also a strong accumulation of activated NK cells and proliferating T cells. So clearly PI3 kinase gamma inhibition had a strong effect on T cell activation and recruitment into the tumors. So Hide then asked whether he could transfer this property to other animals. So he isolated either splenic T cells or B cells from wild type and PI3 kinase gamma knockout animals and implanted them in wild type animals together with tumor cells. So he found that only if you had PI3 kinase gamma knockout T cells, splenic T cells, could you completely transfer this activity to other animals. B cells were not effective in this regard. Then Ryan Shepard in the lab went on to isolate CD8 positive effector memory and central memory T cells from wild type and knockout animals. And he showed that all of the activity, of course, resides in effector memory cells. So our thought is that effector memory cells are trafficking to the tumors, where they're differentiating to active effector cells and preventing tumor regrowth. So we were then able to show that macrophages from knockout animals could stimulate T cells ex vivo. So to do this, we isolated T cells from wild type animals and macrophages from wild type or knockout animals, incubated them together with the E7 peptide in an ELISpot assay, and found then that PI3 kinase gamma knockout macrophages could stimulate wild type T cells by presenting E7 peptide to them. And that was also the case if we took macrophages from IPI 549 treated animals. So finally, we then did T cell receptor sequencing together with Hideyuki Takahashi did this with Darini Murugan and Lisa Cousins at OHSU. And from two different models, and we found that each of the models, there was an increase in the clonal index of the T cell receptors. So that means there's more diverse T cell receptor types that are coming into the knockout tumors. And so there are more unique T cell receptor clonotypes in these animals. And that indicates that there's a beneficial effect on spreading the, on epitope spreading in these tumors. So we've gone on to ask whether IL-12 and interferon gamma play essential roles in the effect of PI3 kinase gamma inhibition. And so we found that if we knock out, that there's a substantial increase in IL-12 in the PI3 kinase gamma knockout tumors. And if we then knock out IL-12, we found that it could completely reverse the beneficial effects of inhibiting PI3 kinase gamma in tumors. So that shows that PI3 kinase gamma inhibition, elevation of IL-12 in macrophages and potentially dendritic cells plays an important role in this process. We then also asked whether interferon gamma receptor was important, as you might expect. Interferon gamma is strongly upregulated when you inhibit PI3 kinase gamma in these tumors. And we had found that the macrophages from tumors appeared to show an interferon gamma-like response. They're activating CXCL9 and 10 and guanylate binding proteins, as well as MHC molecules. And so if we delete interferon gamma receptor from PI3 kinase gamma knockout mice, we also see that reverses the beneficial effects. So that clearly shows us that cytokines and MHC class 2 from myeloid cells activates T cells, and that produces a kind of a chronic upregulation of a good, strong anti-tumor immune response. So what we've shown is that PI3K gamma is upregulated in myeloid cells. It's mostly expressed in myeloid cells. Inhibition suppresses myeloid cell recruitment to inflamed tissues. It also regulates pro-inflammatory and macrophage polarization and thereby inhibits tumor growth, angiogenesis, and metastasis. And it indirectly activates T cells by upregulating MHC class II and producing T cell chemokines. But is this beneficial for cancer patients? So I'm pleased to say that so far it looks that to be the case. Infinity Pharmaceuticals developed eganelisib. This is the small molecule structure, and have shown that it has a preferential ability to bind to and inhibit PI3 kinase gamma over all of the other PI3 kinase isoforms. It also has pretty much no activity on other molecules. So they've done at least 5 clinical trials, and I'm showing you here 4. Mario-1 was a phase 1b study examining the effect of PI3 kinase gamma in checkpoint inhibitor refractory melanoma, head and neck carcinoma, lung carcinoma patients. And I will show you some data from that. They've done a couple of other studies, so I'll show you data from this frontline triple negative breast cancer in combination with anti-PD-L1 and nab-paclitaxel study, and their Mario 275, a study in urothelial cancer in combination with nivolumab. So in Mario 1, they got some data with just treating patients with monotherapy, just PI3 kinase gamma inhibitor alone, and they found that there was a decrease in myeloid-derived suppressor cells, which are MHC class II negative monocytes that are circulating in patients upon 1 cycle of treatment. They also saw an increase in T cell proliferation of PD-1 positive, CD8 positive cells that were in circulation, and an increase in circulating CXCL10. All of these were biomarkers of a potential good response. They also saw increased interferon gamma induced expression of molecules such as PD-L1 and guanylate binding proteins in peripheral blood leukocytes that were surveyed from these patients. So then they went on to do Mario 275, looking at this eganelisib plus nivolumab in urothelial cancer patients, and it was was done in contrast to nivolumab alone patients, and they essentially looked at overall response rates and progression-free survival. So they have now published their 2-year survival studies, and so what they found is substantial improvement in survival in patients that were treated with both drugs versus those treated just with nivolumab. And they did some biomarker analysis of peripheral blood samples from these patients, comparing before and after treatment. And so what they see is that after treatment, a strong upregulation of MHC class II, T cell receptor signaling, class I, antigen processing, and interferon gamma signaling and interferon alpha signaling pathways, suggesting this is working in patients the way we predicted it would work in animals. So the next study was Mario 3, a breast cancer study. This was done simply in comparison to historical controls, but what they did do was they compared PD-L1 negative and PD-L1 positive patient responses. So they observed a tumor reduction in 92.8% of PD-L1 positive patients and 85% in PD-L1 negative patients, shown here. So they actually, these studies are obviously were from a while ago, and I think may have been concluded now, but I don't have the final results. As you can see, though, they had strong tumor suppression results and also had some complete regressions. So they did some analyses of these patients, and here they looked at PD-L1 negative and a PD-L1 positive patient with partial responses, looking at their tissues for the expression of macrophage subsets and T cell subsets. And what you can see is that after treating for 2 months, there's a change. There's less tumor in these sections. And the macrophage types labeled with M2 here have switched to a more pro-inflammatory phenotype. And also, you can see the same thing has occurred here in this PD-L1 positive patient. There's a substantial increase in the number of activated T cells that have entered these tissues as well, or T cells, and that's summarized here. So then they did some gene expression analysis of paired biopsies from pre and post treatment of these patients. And what they observed is a strong upregulation of CXCL9, CCL5, these are all pro-inflammatory factors, also increase in MHC class 2 and 1, and molecules associated with activation of macrophages, and then a downregulation of angiogenesis factors and wound healing factors such as MMPs. They then looked at the pathways that were regulated, and again, they see activation of immune pathways, interferon gamma signaling pathways, and a variety of immune pathways, and a downregulation of basically matrix wound healing type pathways. So these results mirror what we saw in animal models and associate with positive benefits in patients. We've done our own kind of studies like these by doing digital spatial profiling via nanostring methods, in which you incubate tissues with so fluorescent antibodies to identify regions of interest, as well as with either antibodies that are labeled with oligonucleotides or oligonucleotide probes to RNAs, and then we remove those and analyze what's expressed. So here are some examples of fields that we identified. And we were able to find out, and if we look at all the tumor types that were present within this was from the Mario-1 study, where patients were previously treated with eganelisib with anti-PD-1, were then treated with anti-PD-1 and eganelisib. So the changes you see are due to the combinatorial effect of the drugs. And what you can see is, if you look at what's common to all tumors, there's an increase in CXCL10, a variety of pro-inflammatory factors like CXCL9, STAT1, CD40, but there's also an increase in exhaustion markers like PD-1, which is what you would expect if you see a strong increase in inflammatory response. In head and neck cancer, we see some of these same things. IL-10, IL-15 suggests an NK cell response, and head and neck tumors are particularly characterized by NK cells. And we found this molecule, integrin beta 8, which I'll tell you about in a second. And so then in non-small cell lung cancer, we also see evidence of increased T cell activation, but also exhaustion markers. So integrin alpha V beta 8 is a molecule that's been associated with fibrosis. My colleague Dean Shepard at UCSF and his postdoc Amha Atakilit and my postdoc Alice Chen have been collaborating to explore the synergistic effect of combining antibodies to integrin alpha V beta 8 and PI3 kinase gamma inhibitors. Dean has shown that alpha V beta 8 is expressed on regulatory T cells where it controls TGF beta expression. He showed using an antibody he developed to alpha V beta 8 that it could block regulatory T cell expression of TGF beta and strongly synergize with anti-PD-1. Well, we found that it strongly synergizes with PI3 kinase gamma inhibition and wipes tumors out. So we're continuing to explore the mechanism of action, but I believe this is a potential therapy that could be even safer than combinations of checkpoint inhibitors. And as you can see, the tumors are almost all eradicated. And then when we re-challenge them with tumors, they completely eradicate the tumors. So we're continuing to focus on these kinds of combinatorial therapies and exploring mechanisms by which myeloid cells regulate tumors. Here's an example of a study we're doing examining pre and post treatment in collaboration with Lisa Cousins, looking at using multiplex immunohistochemistry to look at the effects of therapeutics on the compendium of immune cells present within tissues. So in conclusion, we've shown that PI3 kinase gamma inhibition can block trafficking of some myeloid cells to tumors, and it can repolarize the rest so that they synergize with checkpoint inhibitors and other T-cell targeted therapies to promote good, strong immune responses in tumors. So I want to thank all the people in my lab who've done this work, including my fine colleagues who came to the AACR with me. Students who just graduated are all shown here, and my lab manager, Mark Paradise, and my postdocs, and my colleagues at UCSD, OHSU, Torino, Salk Institute, UCSF, and Infinity. And I also want to thank my funding sources, which have been important for getting this work done over the years. And then finally, Infinity wants to acknowledge their clinical collaborators and all of the patients who participate in these studies. And then I do want to invite you to join us at the Society for Immunotherapy of Cancer meeting in November in San Diego, where we will be having our first ever totally myeloid cell focused meeting on the November 2nd. So join us, please, and contact me if you'd like any information. So thank you very much for listening, and I look forward to your questions.
Thank you, Judy. That was phenomenal. It's just a wonderful example of bench to bedside, but also true collaboration within your own lab and with others around the world. The problem with this virtual seminar is that you can't actually hear our applause, which would be resounding right now, I'm sure. So thank you. We do have some questions I want to get to. So the first one, well, this may have been addressed, but the question is there evidence that other tumor types likewise show this phenomenon of cancer growth, spread, desmoplasia, and immune suppression based on inhibition of PI3K gamma?
So I guess I'm little stumped by the question. So we have, looked at the inhibition of metastasis and spread in a variety of models, but I think the most telling are the genetically engineered models, the PYMT and the pancreatic cancer models. The only model that I've worked with that's extremely fibrotic is the pancreatic cancer model. We are working now with a genetically engineered lung cancer model that is really interesting, and it doesn't seem to involve a lot of fibrosis, at least not at the stage at which it kills the animals, but it is extremely macrophage rich, and we are currently exploring the effect of PI3 kinase gamma inhibition in that model. All these models require a lot of breeding of animals.
Yeah.
Takes a while.
Yeah. Actually, yeah, I think that question was followed up later. It said, forget my original question, since I now see data for urothelial breast and melanoma. So, but you did expound on that. Thank you. Another question, have you been able to assess the effect of gamma inhibition plus anti-PD-1 in immune excluded, immunologically absent versus immune infiltrated tumor settings? That's interesting.
Well, yeah. So the Lewis lung carcinoma model is, I guess you could call it an immune excluded model. There actually are almost no T cells in it, and there are more NK cells than T cells, but there are also not very many NK cells. It's a highly immune suppressive model. So what we observe when we inhibit PI3 kinase gamma in that model is it doesn't synergize at all with checkpoint inhibitors, which have no activity in it because there are no T cells. So we have a doubling of T cells in it, but it's still not a lot of T cells. So what we did find is that it synergizes well with chemotherapy. And so we still have to go back and combine chemotherapy, checkpoint inhibitor, and PI3 kinase gamma inhibitor, and look at, what kinds of cells are recruited. But currently we're focusing more on the genetically engineered model of lung cancer, which seems to involve a great deal more of alveolar macrophages, at least early on. And so we're trying to understand what role they play, in part in collaboration with Mary Murad, who has made observations about macrophage roles in lung cancer in the past. So there's a lot left to be figured out. But that is a model of immune exclusion where this doesn't work by itself activating a strong T cell response. And so one thing we do observe is that inhibiting PI3 kinase gamma produces factors like nitric oxide that do seem to kill tumor cells. And we've been able to show that we can take supernames from macrophages ex vivo and put them on tumor cells and get some tumor death. And we've been working on, we know part of it's nitric oxide caused, and we also think some of it's caused by other immune factors that we're identifying. So if we do single cell sequencing, we do see a lot of reduction in tumor cells, even when there are not a lot of T cells present. So macrophages can kill as well.
Yeah. Interesting. Yeah, that's really interesting angle. I mean, obviously, good work leads to so many more questions, but you're tackling it phenomenal. So that actually does lead into a couple of other questions. We'll have time for maybe a couple more. Upon combination treatment, anti-PD-1 and gamma, do you see more T cell activation gene expression changes than myeloid gene expression changes, or do you really see it from both compartments? You mentioned the single cell analysis, but
well, I think we see it from both. We have actually done rather limited analysis of the combination therapy. We've been focused more on what PI3 kinase gamma itself does. But by inhibiting just PI3 kinase gamma, we see that there's this particularly strong upregulation of MHC class 2 and chemokines more than anything else, and also TNF alpha by single cell. And then in association with T cells are more activated. They produce more granzyme and more perforin and other factors. We do have to, I think, continue to to address that question. What happens when you put the different therapies together? So, yeah. I think we get a lot more information by doing single cell sequencing. So we're ramping that effort up quite a bit.
Excellent. Yeah, so I think there's just time for one more, and then we'll send you the rest if you want to be able to look at those and answer. So the final question: are there biomarkers? It says in tumor cells, macrophages, T cells. Are there biomarkers that can be used to predict response to the gamma inhibitor?
Yeah,
that's a good question to end on.
So we're actually working on that, but Infinity has, I think, some evidence. So, what I think is promising is we see the same kind of changes in the circulating cells that we see within the tumor. Does that correlate with tumor responses? I actually think it does, but that is something that we have to all work on. So in animals, we see that that's the case, and patients, I think that studies are still going on. So right now there's a clinical trial, a larger scale clinical trial about to start in head and neck cancer. partly at UCSD and other sites that's run by the company, and they will be doing a lot of biomarker studies. So what I guess a biomarker they have observed is that they observe increased PD L1 expression in response to treatments in patient biopsies. So essentially that suggests an interferon gamma response. It's interferon gamma driven, and then they think that makes patients more able to be treated with anti-PD-L1.
Right.
And then, some of the studies we've done suggest upregulation of PD-1 and so in some tumors, integrin alpha V beta 8 suggests that they are biomarkers of response.
Mhm.
But then there are also things that you probably have to inhibit. So I think we're getting there, but I don't think it's complete yet.
Nice. Well, that again, thank you, Dr. Varner, for giving this final seminar in the series. It was phenomenal. Congrats on this beautiful work. And I think I speak on behalf of everyone here. Again, another round of applause for you for your efforts. And with that, we'll sign off. And thank you, everyone, for attending for all these years.
Thank you for the great invitation. Really appreciate it.
Take care.